Ladies and gentlemen, allow me to reiterate what a singular honor and pleasure it is for me to present some of our results and future research efforts to you. This conference means a lot to me. I have been a very enthusiastic participant in all these similar type of conferences, and I was very honored by the invitation to deliver a keynote in the wonderful company of Professor Richard Roberts, Bob Grubbs, Jean-Pierre Savage, and of course Professor Jean-Marie Len. So our efforts are all focused on these phenomenon of reverse pharmacology, observational therapeutics, all of which are geared towards the discovery and development of active new chemical entities. The name of our company is Think, which represents technology, health, innovation, novelty, and quality. The replacement of the K in Think by Q is deliberate. We have to generate all data under very high quality conditions. And I always had a fascination for Mr. Q in James Bond, always trying to discover new technologies. So what are we exactly about? Our stated goals, ambitions, and mission in a way is to establish a new paradigm for drug discovery and development. We are geared and always thinking about how best can we align yesterday, the knowledge derived from the traditional medicines with the modern chemistry and biology of today to deliver effective, efficacious medicine for tomorrow. We are combining the best of the ancient wisdom and particularly the wisdom of Mother Nature, who has been called as the greatest synthetic chemist of all time, who brings together an incredible fusion of all the synthetic procedures. And of course, we have to always utilize the cutting edge science for discovering the medicines of tomorrow. What is our effective starting point? We branch away from pharma's conventional approach, which has always used for their starting point a known and validated biological target with some potential for disease modification. So, of course, people have made many compounds and compound libraries, all of which are geared towards interacting with a chosen biological target. Of course, everyone is focused on defining the human proof of concept, safety, and efficacy, all done through normally accepted and regulatory approved procedures for preclinical and clinical development. Please realize that in traditional pharma, the focus of R&D is overwhelmingly based on establishing safety efficacy, but the important issue is that everybody targets one molecule. There can be only one target for a biological receptor. Some cases people have experimented with some dual function molecules, but not to a tremendously large extent. Then we've had these things like combinatorial chemistry and so on. Now, many of the VCs and other people perceive the drug discovery as a very high risk thing because you always heard these numbers that for every million compound screen, maybe one will make it to the market. So what way are we de-risking things? What is our approach, which is sometimes unconventional, but we use as a starting point these traditional Indian medicinal products with years of safety and efficacy. Our mechanism of action and chemical moieties responsible are not quite known because people haven't invested the effort in determining the chemical structures and the mechanism of action. We are not necessarily relying on the one target, one molecule, but many of our materials are indeed hitting synergistic and dual targets. We hope that since these materials have already been in man, some of the risk regarding safety and some of the risk regarding efficacy could be lowered pending further and detailed experimental studies. Lot of the impetus for our inventions went about with the extremely pioneering work of Dr. Ashok Vaidya and Rama Vaidya. Dr. Ashok Vaidya is known for coining the term of reverse pharmacology, which is defined as a science of integrating bedside documented experiential hits and transforming them into leads by these very seminal exploratory studies. These studies are very detailed, very precise, and one has to understand the biological mechanism of action at each stage. We have to pinpoint the chemical structure of each of the scaffolds that is going to be responsible for the activity. In a lot of the literature, this has been called as in clinico research, in sort of contrast to the in silico, in vitro, and other techniques that have been used. So this tells you a little bit about why we are so optimistic. Our paper has received very many citations, but we are always optimistic and we are hoping that if we use this as a starting point for safe and efficacious medicines, we will be able to reduce the time to market by several years. Please remember, this is not going to be synonymous with cutting corners. You are still going to have to do the studies. I'm only beginning with large data sets likely to be available from this clinical data. Now, ever since I got excited about organic chemistry, it was very clear that natural products was an exactly known thing. It was, in fact, something that deliberately excited many of the chemists. They were the only effective medicines through much of human history. Medieval monasteries always had their herb gardens. And think about it. You saw this literature even in Western monasteries, in Tibetan and Buddhist things, all over India, and so on. And this came to a very dramatic notice of many people. There was a scientist in China, Professor Yu Tu, who was then commissioned by the government of China, headed by Mao Zedong, to treat the scourge of malaria that was prevalent. And Yu and her collaborators, they led a very massive effort, a very massive well-funded effort. They looked at papers dating back to the Han dynasty. They analyzed those papers, then they took soil sample, they cultivated everything, and they got this drug artemisinin into the marketplace. Now you cannot ask for anything better. The story that was always told to us when I was sort of growing up in organic chemistry was the story of the pumas and leopards and all that, who used to have the same shivering, the same chills as was experienced by many people in the villages. And then people started noticing that when these animals congregated around watering holes, drank from that water, some of their chills and all that got reduced. Now, later on, we came to identify this fever as malaria, and later on, people also made the very important observation that this could be due to some of the natural products from the surrounding trees that had been leached into the water. These trees were all cinchona alkaloids, and you all are familiar with the work of Pelletier and Cowan too, who isolated and proved quinine. And then the chemical structure was eventually proven. This was the first total synthesis that I studied in my academic curriculum. And of course, it was the synthesis done by Professor R.B. Woodward, the legendary chemist, and also his colleague, Professor William Von During. Now, this is definitely established that probably King Charles II was cured of malaria, and of course, the curative agent was indeed quinine. Now go to morphine from Papaver somniferum. Frederick Serturner did this. So you have established the fact that natural products were extremely important in the treatment of disease. And this all became, it continued on a very winning trajectory. Very soon, people established the structure of vincristine, vinblastin, podophyllitoxin, camptothecin, Taxol. All these molecules were studied by several brilliant scientists. Many of them did succumb to total synthesis, and there have been some remarkable pieces of research that have been published in the chemical literature. So the concept of using natural products for therapeutic treatment of patients was not new. So why are these natural products being so successful? That first of all, many of them have very complex structures which are endowed with chirality, with diversity. Many of them interact. They are designed by Mother Nature to interact with the enzymes, and many of them are produced for defense and other purposes. Please keep this thing in mind. Now, around the time when I was an active researcher approaching mid-career, suddenly this natural products research started declining in many of the Western companies for many reasons. First of all, the scientists defined natural products as not just being plant-based, but they went into different directions like isolating natural products from mollusks and fungi and bacteria. That was all fine, but it gave you an obvious problem that very soon the commercial availability of that natural product was never going to be sufficient for your studies. Take materials like palytoxin and so on. These were synthesized, subjected to total synthesis in a laboratory after hundreds of steps. Now, you are not able to commercialize those molecules by doing a total synthesis. Another problem came in a lot of the perceived difficulty in securing IP as a monopoly. Many of the pharma companies, of course, had a theory. They would go to countries, they would collect natural products and, of course, patent it, and that will be a monopoly of that particular pharma company. Then the Phuket Declaration was signed, particularly by the BRIC countries, and this did not become a plausible or successful avenue for many of these people. Then we came to combinatorial chemistry, where many people defined the chemical space by these techniques of combinatorial chemistry, not always successful, because very soon people started using the standard reactions like the Keck, which were all designed for synthesis of planar molecules totally devoid of any chirality or diversity. So there was a total absence of diversity in all these building blocks. The newer biology was retrofitted to very mundane, repetitive chemistry of using methyl, ethyl, futile, and so on. And India and China, though everybody wanted to go there, there were no good mechanisms for harnessing the intellectual capital. And many people simply looked upon these countries as low-cost arbitrage for materials. Now, here is my motto: if necessity is the mother of invention, surely frustration is the mother of innovation. All good. We banked on using this Ayush therapeutics. Now, the word Ayush literally translates to life in many Indian languages, and it is a composite of Ayurveda, Yoga, Unani, Siddha, homeopathy. But of course, we don't do so much lab work on yoga and homeopathy, so the word Oz is mainly used in the literature. Now, we have a rich resources which are derived from centuries of work. There are now 300 well documented and in use medicinal plants, all of which have been worked on with a philosophical and rational base. There are many traditional formulations in practice. The CSIR has an experiential database on all these, and a lot of this is hyperlinked to the patent offices in London, Washington, and so on. Now take a look at this particular slide. Many of the restaurants, even in USA, UK, and so on, they offer this Ayurvedic thali. This has been a staple in India, whereby people assemble things in a dinner plate, all of which have some importantly perceived medicinal properties. Take a look at the onion. For example, it is well known that it can be eating raw onions can be good for your health because it can help ameliorate the stress caused by sunstroke and nosebleeds and so on. My grandmother used to give me some pomegranate seeds on breakfast yogurt. Later on, we did come to read that that's a great way of warding off diarrhea and dysentery because of the alkaloids, emetidine and all. But things came to a real head when Harvard and UCLA published a joint paper saying that if you have 2 glasses of pomegranate juice per day, very soon it will regress prostate cancer and have a beneficial effect on breast cancer. Professor Sir Richard Roberts said very nicely that if you do not take food as medicine, very soon you will take medicine as food. Now, we have defined these processes for discovery of an NCE from traditional medicines. Please take a look at that. We are doing some very fundamental studies to establish the safety, to establish the fact that these materials are non-toxic. It is not just a random generation of any data. These are the R&D paths for all these natural products. We do work on the medicinal plants and we subject them to our theories and mechanisms of using reverse pharmacology. This is the information that is typically available. This is gingiber officinale. This is in the Sanskrit language. You see that this is called shunti and all that. Then you have some chemical scaffold information, and then you have some potential therapeutic use for all these materials. Now we have accumulated, there is a large number of scaffolds. There are 10,000 plants that are mentioned in the Indian traditional medicine literature. Clearly, me or anybody else, we cannot work on all of them, but we have identified some early favorites. Take a look at these. These are the turmerones, gingiberones, curcumin, and so on and so forth. So these are what I would call as molecules of incredible preliminary interest for us to develop further. Now, I'm not advocating that we take all these hundreds of molecules, put each one of them in a tablet or capsule, and so on. People frequently ask, what's wrong with the mixture of extracts and all? Absolutely nothing wrong. Once you establish the safety, efficacy, and no toxicity of that, you can go back to administering the extract also. But now it will have a very firm scientific foundation. Take a look at molecules like aldose reductase and particularly focus on things like mangiferin, which have the right structural elements in place for you to do your medicinal chemistry on. Now, what did we do? We looked at a lot of the chemical literature, we scoured it, and we isolated some very useful natural products. And then we decided, well, can we as chemists make these available for larger scale testing? Well, let's focus on a new 10-membered macrolide, which we did, and this had a 10-membered ring lactone structure. Professor Grubbs ventured the opinion that such molecules, it is not so easy to do the complete stereochemical synthesis with defined stereospecificity. So we decided to work in collaboration. We assembled this particular lactone by the well-known Yamaguchi protocols, and then I had a remarkable student, Dhondi Ramesh, and this is what we instructed him to do. He had to deprotect the OPMB protecting groups with DDQ, come up with the unraveled products, and then subject it to the ring closing olefin metathesis. He did, and we were all very, very pleasantly surprised when we got exclusively the Z isomer. Now, this is what is brilliant about some of these students. Dhondi Ramesh on his own essentially went ahead and did the reaction the other way, got some incredible stereospecificity out of it. He got the E isomer. Bob Grubbs decided to do some density functional theory calculations in collaboration with Professor Goddard, and we found something very interesting, and that is that the protecting group itself was directing the course of the reaction as a first example of a protecting group directed olefin metathesis. So this is our synthesis of desmethyl nonenolide, which was giving even more spectacular results. You can see that now we have got both the Z and the E isomer very, very stereospecifically. Then I kept on reading about this new concept of the hybrid molecules. And of course, the natural world inspires us, from artists and philosophers to biologists and chemists. But what does the in a primal sense, what does the word hybrid molecules mean? If you can bring together different structural scaffolds, or even if you can bring different molecules together, you might expect some interesting activity. And this was our first foray into this one. We made the quinolyl chalcones. We could combine the quinoline scaffold and the chalcone into one structural scaffold. And the results were very fascinating for us. Our molecules showed very exciting activity. We could then cyclize this with guanidine carbonate to give us a concave shaped molecule. Now take a look at where we are taking this. Why not combine these molecules together under making these hybrid molecules? That is the guise we are going to use. But take a look at the very profound work of Professor Sharadini Dahanukar. She has worked extensively on this plant called as Tinospora cordifolia. Now, around this time, lot of the anti-diabetic medications in the Western world were being withdrawn for cardiovascular risk. We decided that Mother Nature has made these leaves heart-shaped. They were defined and speculated on in literature role of having a cardiotonic effect. Wow, isn't that nice? So we decided we are going to separate the actives from here, make hybrids with pioglitazone and some of the diabetic medications, and we were successful in doing that. We have coupled berberine and we have coupled some of the syringine and all that. One of the fascinating molecules from Tinospora is this octacosanol. And please notice also the polysaccharide structures. Now enters a remarkable scientist. He formerly worked for Professor R.B. Woodward. He's Anton Fleury, who did remarkably well at Pfizer, but he has finally found out a remarkable computer program which is called as informational flow analysis. It really offers a very exciting level of chemoinformatics. They can analyze data across trillions of data points. They can scan, analyze all the structures inherent in most of the plants in the world, and then draw some defined correlations. Go to the country of South Africa, which started work on this particular project. Now, in sub-Saharan Africa, and Namibia, and so on and so forth, many of the locals used to chew on the glabrous roots of this plant. When they're tired, famished, hungry, far from home, they would chew on these particular plants. And why would they do that? It was found out that their appetites were suppressed to the point where they didn't feel so hungry. At least it gave them time to reach home. Now, they defined this as a new mechanism of satiety. So, of course, Pfizer got very interested in the project to the point where everybody was looking for an anti-obesity drug. So they isolated the steroidal glycosides. Professor Stephen Lay's technology was used to synthesize some of these sugars and the steroidal aglycone, which was a very fascinating discovery. But here you came face to face with reality. And what was the reality? The reality very simply was that this plant grows only 6 inches in 12 years. Now that does not do wonders for the sustainability, that you have to wait forever, and you cannot simply introduce these materials into the marketplace by doing a total synthesis of these incredibly complex molecules. Now here comes Anthony Fleury's program, where he suggested to us that in India itself, in many states, you have a plant called as Caralluma fimbriata, which is also an appetite suppressant, which has similar sugars, which has similar steroidal glycones. But ladies and gentlemen, this plant grows 12 inches a month. Now, isn't that a step in the right direction? And we are following this up to a very big extent. On the road, we have encountered some very fun things, like this is the work of Professor J.S. Yadav, former director of IICT. He reduced a ketone to an epimeric mixture of alcohols by using Daucus carota. That is nothing but carrots. And he was able to put this in place. Then another pharma company got excited about this, told us to use this method, which is green chemistry, On their ketone, well, they gave us 200 kilos of material. We would have needed about 3 tons of carrots. When our people went to buy these, they were visited by the local police because next day there was a festival at which the sweet that was made was typically carrot cakes and halwa. And people speculated that these are bad guys who are trying to corner the market and drive up the price. And even more amusement to me was provided by this particular compound, which is a des aza-desferrithioceine molecule isolated from natural sources. This was the work of Ray Bergeron in the University of Florida, Gainesville. And as this drug advanced, it was clear that one of the key raw materials was going to be cysteine. Now, I got a call from the pharma company saying that, doctor, we have now identified a source for this in the USA. It is about 225 to 240 a kilo, and surely in India people are selling it for $3 to $4 a kilo. Then the address of that plant was right near a major temple complex, and then it suddenly struck us that many devotees go ceremoniously have a haircut, which is very ceremonial. Then somebody figured out that you can take this hair and hydrolyze all that with concentrated HCl to offer beautifully crystalline cysteine hydrochloride. Well, everybody got excited. The FDA came to visit this facility, and some Orthodox Jewish gentleman from New York then enlightened us, saying that the cysteine is also used by them for getting kosher certifications, and the hair are also exported for wigs and so on. So, all told, a very fascinating and fun visit. Now enters an extraordinarily brilliant Professor Martin Burke. I would call him the enfant terrible of modern science. He conceived of the idea of the natural productome. He has always preached that we chemists need to coalesce around a big idea. The physicists did that for the Higgs boson. The biologists did that for the human genome. Why can't we do it natural productome? He has now devised a lot of sophisticated computer programs for which he can prove that most of the natural products existing in the universe can be made up of some 12, 1400, whatever it is, basic fragments. And can we combine them by using the power of synthetic chemistry, which will be like a moonshot for chemistry? Of course, they have published this work. He is also known for the invention of this magnificent synthesis machine, which is a true revolution in a sense, and that's how they named the company. But you can do all kinds of nice and fancy things with it. You can take different building blocks and different scaffolds, and you can make some small molecules. Please notice that some of these techniques are well known to all of us synthetic chemists, but he has brought together in an extraordinarily brilliant fashion. Now, for the last couple of minutes, may I tell you about the inventions of things going on in other areas as well. This is Nirvana water. Now, Nirvana is a very sacred thing for people, especially from the Hindu, Buddhist, and other cultures. But what have they done? They have promoted an active lifestyle brand with a proprietary patented line of infused materials. How nice is that? This draws on muscle wellness, and over the years, many of these athletes who were doing some incredible levels of activity, but sometimes they would strain their muscles, they would stress them, and they needed some muscle rejuvenation, they needed some muscle healing and muscle cure. So Professor Naji Abumrad, an extraordinarily distinguished scientist from Tennessee, he made a derivative of lysine. It was a metabolite. It was called as beta hydroxy beta methyl butyrate, and made it as a calcium salt and offered it as a tablet for the healing of the muscles and so on. Now, all this is very good, but then people figured out that as we get older, above 40, our muscle strength diminishes by 1% every year. So, can this discovery be made available to the lay person? Why do only athletes have to benefit from this wonderful technology? Now, fast forward to near Utica, New York, people had bought some remarkable land, 1700 acres in the Adirondack wilderness. Now, below that, there is two ice age aquifers. There are two of them containing trillions of gallons of water. There are 5 natural springs over there. Now imagine the possibilities. You have a bottling plant which will bottle lot of this water and send it. Well, that is exactly what is happening. This water is infused with HMB and is now available in stores in the USA, and very soon it is hoped that it will be available in other countries as well. Now, what is the application of this to traditional medicine? Well, it can be sold as medicated water. We can infuse things like tulsi extract, we can infuse Tinospora cordifolia, and so on. In conclusion, may I state that I am absolutely convinced that these traditional systems of medicine, which are founded on an integrative systems theory approach. This is a tremendous boon to all of us. These things can serve as a very rich reservoir of natural products. They offer organized, systematic, predictive, safe, cost-saving measures. And we ultimately owe it to ourselves to study these natural products which Mother Nature has endowed upon us with her infinite bounty. These are products with holistic harmony and reductionist reliability. So let us jointly harness this transforming power of modern chemistry and biology. Allow me once again to reiterate my pleasure, my gratitude to the organizers for having invited me to this extremely prestigious forum. And I close with my thanks to all of you for your kindness, your patience, and hopefully your encouragement. Thank you.